Today's session at Medical Sciences, we want to discuss about fixatives, which we use in histopathology lab. So we want to see how tissues are fixed in the laboratory or in histopathology lab when we want to rule out tumors or cancerous cells. And there are different types of fixatives. So in this session, we are going to look at what is a fixative. We are going to look at the principle behind the fixatives. Then we shall look at the aims or the significance of fixing samples. Then we shall look at lastly classification of fixatives. So after doing that, we are going to begin with what is a fixative. And a fixative is a chemical. They are chemical or physical substances which are used to preserve the morphology and structural details of the cell. Because we see after the tissues have been removed from the living organism, there is what we call postmortem changes that occur on them. So when we fix them using these chemicals, they preserve the cellular components in their lifelike state. So fixatives are chemicals or physical components that preserve tissue or cell components in their lifelike state, or preserving them to maintain their morphology and structural details. So that is what we can call it fixative. And this fixative, they work under the principle whereby the principle of fixatives, we are going to see that they some act by formation of cross-linkage, because cross-linkage of proteins, to form a three-dimensional structure. And when cross-linkage occurs, for example, when you're using formaldehyde, glutaraldehyde, they form cross-linkage of proteins in the cell, which ends up forming a mesh, what we can call a wire mesh. And this wire mesh is the one that keeps the structure of the tissue or cells in their position. So this mesh, wire mesh which is formed, is the one that keeps the structure in their position. They keep the cellular components in position. That is what one way of the principle of which is majorly for formaldehyde and glutaraldehyde plus osmium tetraoxide, whereas other fixatives act by coagulating. They cause coagulation of proteins. For example, alcohols, acetone, alcohols like methanol, like absolute ethanol, they cause coagulation of proteins. And when proteins coagulate, they form a semi-solid structure which keeps the cells in their position. And others act by precipitating proteins in the cell. Precipitation of proteins in the cell. So this is the mechanism of action of fixatives. So after these fixatives, so the major principle behind is by formation of cross-linkages or coagulation or precipitation of proteins, which keeps the cellular components in their position by formation of these meshes, hence preserving the morphology and structural details of the cell. And this one is the one that keeps the cells as much as possible in their lifelike state. So after that, we are going to see what is the aim of fixatives. One of the aim of fixatives is to preserve the cell morphology Then another importance or aim of fixing tissues is to prevent postmortem changes. Prevent postmortem changes that occur on tissues after being cut or biopsies And postmortem changes include autolysis. That is what we call autolysis or autolysis, and putrefaction. These are the two postmortem changes that occur on a tissue after it is cut from the living body. Whereby autolysis, here it is by endogenous enzymes like lysosomes. This is due to action of endogenous enzymes like lysosomes, which break the cells. So we are seeing these enzymes found in the tissue, endogenous enzymes like lysosomes. They cause cell lysis or break down cells, leading to damage in the morphology. Then putrefaction, this is due to action of bacteria. So these fixatives should be able to kill the bacteria, which may cause putrefaction. So this is another second aim of fixing. Then another one, number 3, we can look at they are used to convert cells which are in a semi-liquid or semi-fluid state to semi-solid. Why are we converting from semi-fluid state to semi-solid state? Remember, this tissue, we want to do many tests, or it is going to be maneuvered through different processes. So it renders them rigid, or they form semi-solid state, so that during tissue processing, they remain with their integrity without tissue damage. That is what we call converting them from semi-liquid state to semi-solid state. So this one causes hardening of the tissue, so that during processing, they do not be damaged. And another one is that these fixatives, some of them enhance staining. They enhance staining. So this is another significance of fixing tissues. Then another one is that some fixatives increase refractive index. They increase refractive index of indices of the tissues or the cell components, hence easing their visualization under microscopy. So these are some of the aims of fixing samples. After looking at the purpose of fixing, now we go directly to classification of fixatives, whereby fixatives are classified under two classes, whereby class 1 is physical fixatives, then B is chemical fixatives. Then physical fixatives, here we use either heat, but which is not commonly used in histopathology lab, and freezing. Freezing is done on frozen sections. Then chemical fixatives are the one we are going to expound in details in this session. So we are going to explain more on classification of chemical fixatives, because we have seen physical fixatives are not commonly used like heat. Maybe freezing is the one that is used for forming frozen sectioning, whereby they are freezed in negative degrees, like -80. Here the temperature, we use -80 degrees for IHC, that is immunohistochemistry and histochemistry. So after this, we are going to now to go in and expound or explicate more on chemical fixatives. So chemical fixatives, they are further classified into two, whereby we have simple fixatives and compound fixatives. We have simple and compound. What do we mean by simple? This one is made up of only one chemical compound, whereas chemical fixative, whereas compound fixatives, they are composed of 2 or more simple fixatives. And under compound fixatives, we are still seeing, we are categorizing them further into 3, whereby we shall have nuclear fixatives, we shall have cytoplasmic fixatives, and histochemical fixatives. And under physical fixatives, we have examples like formaldehyde, is an example of a simple fixative. Another one we shall have glutaroaldehyde. Then we have alcohol, we have acetone, then we have osmium tetraoxide, then we have picric acid, then lastly we have mercuric chloride as an example of simple fixative. Then we have potassium diachromate. Then lastly we can have acetic acid. So these are examples of simple fixatives. And formaldehyde, which causes, and glutaroaldehyde, which cause cross-linkage and formation of methylene bridges that lead to the formation of wire mesh that holds the tissue in place by forming the wire mesh. For them, formalin comes in concentration of 37 to 40 degrees to 40%, and this is during preparation. During fixing, we normally use 10% formalin or neutral formalin. 10% formalin is the one which is commonly used. So during preparation, we take this 37 to 40 as 100%. But in case you are given a figure below 37, then you take that actual figure. Then it comes in gaseous form but soluble in water. So this 37 to 40 is in gaseous form. So during preparation, using RV/O, we take this one as 100 so long as you are given between 37 to 40. But below 37, you use it as an actual number. If it is 35%, you use 35% as the original concentration. But when it is between 37 to 40, you use it as 100% as original concentration to prepare 10% formalin, which is a simple fixative. And this one is routinely used but it does not preserve carbohydrates, but routinely used for preserving other tissue components. And when formalin is stored in a cold place, it can lead to formation of paraformaldehyde, which are seen as white precipitates within the formalin. So whenever they are formed, we can easily remove them by filtering. And another thing is, when it has stayed longer, that is this 10% formalin can form yellow pigments, what we call formalin pigments, so which can affect the staining when they are present. Then we can see glutaroaldehyde also does the same by cross-linkage. Then alcohols used, we use either absolute methanol, absolute ethanol, we use methanol. These are some of, this one you can call it ethyl alcohol. It's also a fixative, and this one acts by coagulating proteins. And this alcohol is not used alone. This one is used majorly in canoes fluid. Canoes fluid, we use alcohol. Then acetone is best for frozen sections, for sections or for samples which we want to do histochemistry. If we are interested in doing histochemistry, then we preserve our samples using cold acetone is best, because it preserves the cell, the chemicals, the enzymes within the cell. Then osmium tetraoxide is also a simple fixative plus picric acid, and this picric acid is used in Boon's fluid. It is not used alone because it causes shrinkage of cells. Then mercuric chloride is also used in Zenker's fluid, but it is not used alone because of its shrinkage effect. So we are seeing picric acid and mercuric chloride cannot be applied alone, but they are used in combination like in the bones fluid and Zenka, because they cause cell shrinkage. So to prevent cell shrinkage, we have to put it in combination with others like glacioacetic acid, which causes swelling, cell swelling, to counteract the effect of shrinkage. So acetic acid for it causes cell swelling. Then we have also potassium dichromate, and this potassium dichromate is the best fixative for mitochondria, for preserving mitochondria and glycogen. So mitochondria, if we want interested in preserving mitochondria, we can use potassium dichromate as a fixative together with the phospholipids. If we want to preserve mitochondria and phospholipids, we can use potassium dichromate. So these are the simple fixatives we can discuss. Here we have looked at formaldehyde, glutaraldehyde, alcohols, acetone, osmium tetraoxide, picric acid in the bones fluid, mercuric chloride, potassium dichromate, and acetic acid, and we have seen what they can be used for. Whereby, don't forget, for carbohydrate we don't use formalin, and even alcohol, because they do not well preserve the carbohydrates. About osmium tetraoxide, it also causes cross-linkage as formaldehyde. Then after finishing this, we can talk about nuclear stains, nuclear fixatives. So these fixatives, for them, they preserve the morphology of the nucleus. But before we look at the nuclear compound fixatives, there are others which we call microanatomical. So the first one, we have what we call microanatomical fixatives. Then here we shall have what we call cytological. And under cytological, that's where we find nuclear and cytoplasmic. Here I'm going to list them, then we shall expound them as we move on. So under microanatomic fixatives, we shall see examples like Zenka, Zenka's fluid, we shall have Zenka formal, we shall have Boons fluid. These ones are part of microanatomical fixatives. Then we shall have Gender's fluid. So these are four types of fixatives under microanatomical. These ones preserve the cell morphology specifically. The Zenka's fluid, the Zenka's formal, Boons fluid, and Gender's fluid. Whereas cytological, we have the nuclear ones, and under nuclear we shall see examples of nuclear fixatives. We have we have newcomers. Newcomers fixative is one of the nuclear fixatives. Then others we can have under nuclear, we shall see example of Canoe's fluid, and Clark. So these are fluids which are found under nuclear. Then cytoplasmic, we shall see only Champy's fluid and formosali. These are the ones we can list under here, under cytoplasmic. Then under histochemical, we shall see only cold acetone being an example of a histochemical. Here we can put cold acetone, which can preserve frozen sections for histochemistry. So we are going to talk about we shall talk about microanatomical, we shall talk about nuclear and cytoplasmic fixatives. So to begin with, we are going to look at microanatomical fixatives, whereby under microanatomic, that is part of compound. So under compound fixatives, we're going to look at microanatomic fixatives, whereby we have seen we have Zenker's fluid, we have Zenker's formal, then we have bones, then we shall look at gender. So all of these ones, they act by preserving the anatomy or the structural morphology of cells. And this Zenker's fluid, as we said in the beginning, it contains picric acid, the Zenker contains mercuric chloride, contains potassium dichromate, it contains sodium sulfate. So after combining mercuric chloride, potassium dichromate, and sodium sulfate, those are the one ingredients together with distilled water. It's the one we use to prepare this Zenker's fluid. But when it is going to be applied, we add drops. So on application, that's when we add drops of glacioacetic acid. So glacioacetic acid is added in the Zenker's fluid when we want to stain. We put drops of glacioacetic acid to stabilize the Zenker's fluid. Whereas the difference between Zenker's fluid and Zenker's formal, another name for Zenker's formal, we can call it Halley's fluid. This one it is the same combination of Zenker, except on application, instead of adding glacioacetic acid, here we add formalin. So that is the only difference between Zenker's fluid and Zenker's formal. And Zenker's fluid is used for routine samples, for routine histological tests, whereas Zenker's formal is good, or it is best used for fixing. This one, Zenker's formal, can be used for fixing samples like bone marrow, soft samples like bone marrow, can fix spleen, and organs that contain much blood. Organs that contain blood like liver, they are best fixed using Zenker's formal, whereas this is for routine, Zenker's fluid. Then bones, this bones, for it contains picric acid. It's the one that contains picric acid plus alcohol. So this bones fluid, for it is good for testicular samples and intestinal or GIT biopsies. And even when we are looking at fertility tests for sperms, the fertility tests are these ones preserve well the nucleus or the sperms. So if you want to rule out oligospermia, azoospermia, you can preserve the testicular tissues using bones fluid. Then gender's fluid is best for fixing or for preserving glycogen. So these are all multiple choice objectives, so which you need to know that if they ask you which of the following compound fixative is used for preserving bone marrow tissues, spleen, or tissues with much blood, that is Zenker's form. If they ask you which of the following is used for preserving testicular or GIT biopsies, that is Bone's fluid which contains picric acid and glacioacetic acid together with chlorophyll. Then gender's fluid, for it is for glycogen. So it is very important to understand the function of each, apart from knowing that they are working at the principle of preserving the cell morphology or the anatomy. So as we continue, we are going to look at also cytological fixatives. We are done with microanatomical fixatives, which is part of compound fixatives. This was compound. We are still on compound where we began with microanatomical. So now we are going to look at cytological. And under cytological, we said number one is nuclear fixatives. We have nuclear fixatives. These ones preserve the nucleus of the cell. And among the nucleus fixatives, we have seen examples like canoid fluid, we have seen example of Clark fluid and newcomers. So these are the three examples of nuclear fixatives which preserve the nucleus of the cell for further studies. And this canoid fluid, these ones majorly contain alcohol, absolute alcohol, they contain chloroform, and glacioacetic acid. This is the composition of canoid fluid. This canoid fluid is the one responsible for preserving the nucleus of cells. And apart from preserving the nucleus of cells, this canoid fluid is a good fixative for carbohydrates. So if you want to preserve carbohydrates and you work for histochemistry, you use canoid fluid for carbohydrate and glycogen. Whereas Clark is best used for preserving chromosomes and mucopolysaccharides in the tissues for histochemistry. Then newcomers fluid is best for preserving, this one is for preserving majorly cell cultures. You can use it for preserving, this one is better for cell cultures. On a slide, when you have prepared cells in a cytological lab, and you have come up with the cells on the slide, those cell cultures and mucopolysaccharide are best fixed by Clark's fluid. Then this newcomers is for chromosome preservation. So if you want to preserve the chromosomes for visualization, you can easily preserve them using newcomers fluid. And sometimes also mucopolysaccharide. So mucopolysaccharides are also preserved using newcomers. So this is what we can discuss about nuclear staining under cytological fixatives, whereby we have seen the canoid fluid is good for carbohydrate and glycogen. We have seen this Clark good for cell cultures, and at the smaller extent mucopolysaccharides, but here we can remove it and we put chromosomes plus chromosomes. Then we have seen newcomer good for mucopolysaccharide and chromosomes. So in the objectives or multiple choice questions, they can easily ask you that which of the following cytological stain is used for preserving mucopolysaccharide, that is newcomer. Then for preserving cell cultures on the slide, that is lacks fluid, whereas the fixative used for preserving carbohydrate and glycogen, which is part of carbohydrate, that is canoized fluid, is the best. Then lastly, we can talk about cytoplasmic. Cytoplasmic cytological fixatives, and under cytoplasmic we have seen majorly Champe's fluid and formal saline. And this Champe's fluid, for it as a cytoplasmic stain fixative, it fixes or it preserves the cytoplasm of cells. So when you prepare cytological specimens to preserve their cytoplasm, it is better using this Champe's fluid. And this Champe's fluid is good for preserving what we call mitochondrial fats. This one is good for mitochondrial fats. Or you can call them lipids. So they are best preserved. So when you're doing histochemistry and you are interested in demonstrating presence of fats or lipids in the mitochondria, you use Champe's fluid. Then formal saline is also good for maintaining and preserving the cytoplasm of cells. You can either use the formal saline or formal calcium. If you don't have formal saline, you can use formal calcium to preserve cytoplasm of cells. So in case they ask you in a multiple choice question, because fixatives are majorly asked under multiple choice questions, if they ask you which of the following compound fixative is used for preserving mitochondrial fats or lipids, that one is Champe's fluid. Then lastly, histochemical, we said majorly we have cold acetone, and we also have vapor fixatives, which we shall see them under histochemistry. So thank you so much for listening, whereby we are concluding by seeing that classification of chemical fixatives, we have what we call simple and compound. We have given examples of simple fixatives and their significance. From there, under compound, which has more than one, we have seen they are categorized further into three, that is microanatomical, cytological, and histochemical. And under cytological, we further categorize them into nuclear and cytoplasmic, whereby under nuclear we have seen newcomers, canoids, and Clark, whereby newcomer we have seen it is good for mucopolysaccharide and chromosome, canoids being good for demonstrating carbohydrates, then Clark good for chromosome and cell cultures. Then we have seen Champe is good for mitochondrial fats or lipids preservation. Then formal saline also for cytoplasmic structures of the cell. Then we have ended with the cold acetone for frozen sections. Thank you so much for listening at Medical Sciences, where we are always privileged, and we always remind you to subscribe. If you are interested in supporting this work, you can join and become a member by paying 10,000 per month to support this work so that we can continue serving the community. May God bless you.